Hello and welcome to ResearchPod. Thank you for listening and joining us today. Our immune system can be affected by many different types of disorders that impair its function, including autoimmune diseases, such as systemic lupus erythematosus or SLE. Unfortunately, we still don't fully understand the cellular and molecular mechanisms behind this disease. In this episode, we look at the research of Professor Shunichi Shiozawa and his colleagues at Kobe University and the Institute for Rheumatic Diseases in Japan, who since the early 1980s have focused on unlocking the secrets of autoimmunity with extensive experimentation. The immune system has two principal lines of defense: innate immunity and adaptive immunity. First, innate immunity provides a rapid and nonspecific immune response against microbial aggression, whereas adaptive immunity eliminates specific pathogens or pathogen-infected cells and provides the basis for protection from infectious diseases. Adaptive immunity remembers pathogens the body encountered previously and can respond to them more rapidly and effectively. Most important, however, is the coordination between the two systems for the efficient recognition and clearance of pathogens. During adaptive immune responses, specific T and B lymphocytes become activated. T lymphocytes derive their name from the thymus, a small gland in the lymphatic system, where they become specialized. B lymphocytes differentiate in the bone marrow. All T lymphocytes are able to recognize and bind to foreign antigenic proteins presented by major histocompatibility complex, or MHC, molecules. MHC in humans is called human leukocyte antigen, or HLA for short. T-cell receptors, or TCRs, have an extremely diverse repertoire from individual to individual, which is required for the recognition of a wide range of foreign antigens. Such a diverse TCR repertoire is created through a random generation process called V, or variable, D, diversity, and J, joining gene recombination, where gene segments are reassembled by a cut-and-paste mechanism during T-cell development. This VDJ recombination, also called TCR revision, is initiated by recombination activating gene, known as RAG complexes, whose expression is restricted to developing T lymphocytes in the thymus. T lymphocytes that have autoreactive TCRs are eliminated, and the TCR revision does not occur after the T cells have left the thymus, because RAG is downregulated by the steady-state mutual interaction between mature T cells. These are the safeguarding mechanisms inhibiting autoimmunity. Based on their surface markers, T lymphocytes are primarily subclassified as cytotoxic T lymphocytes, or CD8+, and helper T lymphocytes, or CD4+. While cytotoxic T cells are critical for the clearance of target cells such as virus-infected cells and cancer-transformed cells, B cells participate in the production of the various circulating antibodies in response to a variety of foreign antigens. On the other hand, helper T cells interact with the aforementioned cells to regulate these cell-mediated and antibody-mediated immune functions. Specifically, a subset of helper T cells, T follicular helper cells, have a profound effect on antibody production by providing the activated B cells mainly in lymph nodes and spleen with growth and survival signals for their subsequent differentiation into long-lived antibody-producing cells. Dysfunctional T and B cells have been shown to be harmful players in autoimmune diseases, causing the immune system to react against itself. Moreover, although cooperation between T cells and B cells ensures successful immune function, the interaction between these cells is also implicated in the development of autoimmunity. SLE is a multisystemic autoimmune disease affecting multiple organs and tissues such as the brain, blood, and kidney in most patients. Improvements in treatment with a smaller use of immunosuppressive drugs and more efficient treatment of infections have significantly increased the 10-year survival rate to more than 70% over the last 50 years. Nevertheless, current management strategies show limited benefits. Corticosteroid therapy, for example, is frequently associated with long-term adverse effects. Originally, lupus was considered a skin disorder, The word lupus is derived from the Latin word for wolf and was used to define the skin lesions, which resembled a wolf's bite. Later, it was realized that lupus erythematosus is a systemic disease, characterized by defects in immune responses causing inflammation and damage in various organs. In 1949, researchers McKay and Burnett proposed their autoimmune disease theory, which suggested that immunologically competent cells or antibodies attack the normal components of the body. Although this initiated extensive research since, the precise mechanisms of SLE were still not completely understood. Substantial research focused on the abnormalities of pathogens, including autoantigens, as the cause of systemic autoimmunity. Yet the pathogenesis, or the mechanism of development of SLE, remained a puzzle. In 2009, Shiozawa and colleagues described a novel approach to researching the pathogenesis of autoimmunity. Unlike previous studies, they investigated the integrity of the immune system. The researchers overstimulated the immune system in mice that are not prone to autoimmune diseases by exposing them repeatedly to an antigen and successfully induced the development of SLE. In other words, the overstimulation of the immune system by external disturbance disintegrated the steady-state immune response by surpassing its stability limit, ultimately leading to systemic autoimmunity. Based on their results, Shiozawa and colleagues proposed the self-organized criticality theory to explain the pathogenesis of SLE. According to this theory, autoimmunity does not occur when the immune system is stable and its integrity is maintained. However, when repeated immunization with an antigen surpasses the stability limit of the immune system, also known as self-organized criticality, systemic autoimmunity takes place. Another important finding is that no specific antigen is required for autoimmunity to take place. Furthermore, Shiozawa and colleagues identified novel T lymphocyte clones that are responsible for the induction of SLE. They demonstrated that repeated exposure to an antigen induces the development of autoreactive T lymphocyte clones through the revision of their TCRs. These novel T cells, which Shiozawa named autoantibody-inducing CD4 T cell, or AICD4 T cell, have been shown to be capable of stimulating both cytotoxic T cells and B cells. Thus, AICD4 T cells direct B cells to secrete autoantibodies, including anti-DNA antibodies, which are a characteristic of lupus. In addition, AICD4 T cells stimulate the maturation of cytotoxic T cells into effector or fighter cells that are required for their cytotoxic actions and tissue injury. SLE is characterized by various cellular and molecular abnormalities, including the formation and deposition of autoantibodies and antigen-antibody complexes, named immune complexes, known as ICs in various organs. In 2013, following the identification of the AICD4 T cell in the pathogenesis of SLE, Shiozawa and colleagues further investigated the mechanisms of autoimmune tissue injury in the kidney with a specific focus on the effector T lymphocytes. The results showed that autoimmune tissue injury is caused by the cytotoxic T cells, which matured into effector cells with the help of the AICD4 T cells. These cytotoxic T cells recognized antigen presented as IC on the target tissue and induced renal disease. In their latest work, Shiozawa and colleagues paid specific attention to the AICD4 T cell, which is directly linked to the synthesis of diverse autoantibodies by activated B cells and the maturation of effector cells during autoimmune tissue injury. They isolated and characterized this key player to uncover the mysteries of SLE pathogenesis, as well as to propose a potential therapeutic target. The molecular and biochemical analysis demonstrated that AICD4 T cells are T follicular helper or TFH cells, and uniquely express the guanine nucleotide exchange factor DOC8 on the cell surface, now called DOC8 plus TFH cells. The TFH cell is a principal conductor of the immune response, directing antibody formation by B cells and regulating cellular cytotoxic activity. The transfer of these DOC8 plus TFH cells to healthy recipient mice resulted in the generation of a variety of autoantibodies and organ diseases, such as kidney disease. It also led to the generation of lesions in organs other than kidney, primarily skin, lung, and spleen, showing that these cells initiate SLE. The key finding is that the TCR of DOCK8 plus TFH cells are newly generated. This results in novel autoreactive T lymphocyte clones which induce SLE. Shiozawa showed that when the steady-state response is disrupted, that is to say, the T-cell response surpasses the immune system's self-organized criticality, RAG gene complex initiating the VDJ recombination is re-expressed. In DOCK8 plus T follicular helper cells, TCR gene chromatin becomes accessible to transcription factors, such as E22 or DOCK8, and novel TCRs are generated. Thus, DOCK8 plus TFH cell is a novel T follicular helper cell, which underwent TCR revision in the splenic red pulp and became an autoreactive T cell with the capability to initiate the production of a variety of autoantibodies and the development of SLE. They also found that these novel DOCK8 plus TFH cells are significantly increased in the spleen and peripheral blood of patients with active SLE. Splenic red pulp is the anatomical location where varieties of antigen can come in and also abundant in the antigen-presenting dendritic cells that are capable of cross-presenting such antigens to cytotoxic T cells. This means that the antigens coming in can smoothly stimulate and mature cytotoxic T cells responsible for lupus tissue injury. Living organisms are continuously exposed to various pathogens, including non-fatal viruses, which can infect the host if they evade the cytotoxic T cell responses. An example of continuous exposure to pathogens might be the measles virus infection, associated with the recent outbreak in Japan in a subpopulation of young adults who avoided vaccination against the virus. The COVID-19 virus may be another example. The virus may not induce an apparent immune response due to the low levels of cytotoxic T cells in memory at the time of the reinfection. On the other hand, repeated exposure to such an antigen in combination with appropriate host HLA may overdrive the immune response to surpass the immune system's self-organized criticality, thereby leading to the development of the novel DOCK8 plus T follicular helper cells, which then induced SLE by the production of diverse autoantibodies and the cell-mediated response. Most recently, Shiozawa and colleagues have presented a potential SLE therapy in their latest research. They demonstrated decreased production of autoantibodies, improved kidney histology, and reduced lesions of the skin, lungs, and thyroid following the treatment with DOCK8 antibody targeting the DOCK8 plus T follicular helper cells, the key players in SLE pathogenesis. Thus, Shiozawa and colleagues' years of dedicated work offers new insight into the mechanisms of systemic autoimmunity with the presentation of the self-organized criticality theory. Identification and characterization of the DOCK8 plus T follicular helper cells not only shed light on the SLE pathogenesis, but also revealed a possible target of SLE cell therapies. That's all for this episode. Thanks for listening. The links to the original research papers can be found in the show notes for this episode. And as always, stay subscribed to ResearchPod for more of the latest science. See you again soon!